Good afternoon, everyone. My name is Giovanna Guerrero Medina, and I'm the Director for Diversity, Equity, and Inclusion at the Yale University and Director of the Yale Ciencia Initiative at Yale School of Medicine, and one of two co-chairs for Intersection Science Fellow Symposium. I hope you have been enjoying the Fellows and Associate talks and the sessions organized by our postdoctoral leaders. We have come to our second keynote of the symposium, offered by a scientist who exemplifies the values of our initiative. Dr. Huda Sogbi is a clinician scientist who studies the molecular mechanisms of neurodevelopment and neuroregeneration. She was born in Lebanon and attended the American University of Beirut until 1976, when civil war broke out. She then moved to the United States and obtained her medical degree from Meharry Medical College. She completed her training in pediatric neurology at Baylor College of Medicine and then a postdoctoral fellowship in molecular genetics. In 1985, she established her own research laboratory, focusing on identifying the genes and mechanisms underlying brain development and degeneration. Today, Dr. Sogbi is Professor of Molecular and Human Genetics, Pediatrics, Neurology, and Neuroscience at Baylor, Director of the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital, and a Howard Hughes medical investigator. Dr. Sogbi's interests range from neurodevelopment to neurodegeneration. Her discovery with Harry Ward of spinocerebellar ataxia type 1 is caused by expansion of a polyglutamine tract, and her subsequent studies that such expansion leads to accumulation of the mutant protein in neurons has had profound ramifications, since many late onset neurological disorders involve similar accumulations of disease-driving proteins. Dr. Sogbi's work in neurodevelopment also led to the discovery of the gene MATH1 ATOH1, demonstrating that it governs the development of components of the proprioceptive valence, hearing, vestibular, and breathing pathways. And her group also discovered that mutations in MECP2 cause the postnatal neurological disorder Rett syndrome, and revealed the importance of this gene for various neuropsychiatric features. Her work has been recognized through numerous honors and awards, including the Vilsack Prize for Biomedical Research, the Gruber Prize in Neuroscience, the Breakthrough Prize in Life Science, and the Brain Prize. And she is an elected member of the US National Academy of Sciences and the National Academy of Inventors. But perhaps most important to this event, Dr. Sogbi is very well known for her mentorship. She has trained over 90 scientists and physician scientists. When we approached her with the invitation to speak at our event, Dr. Sogbi responded almost immediately. Instead of us accepting our honorarium, she asked if we could make a donation instead to the Sogbi Mentorship Fund, an endowment that she established to support postdoctoral fellows who are aspiring faculty to generate preliminary data for bold new ideas to pursue in their labs. After Dr. Sogbi's keynote, questions will be moderated by Dr. Sohini Banerjee and Dr. Maiko Kitaoka, who, in addition to being talented scientists, are the co-chairs of the ISFS Programming Subcommittee. So please join me in welcoming Dr. Sogbi.
Thank you so much, Giovanna. This is such a generous and kind introduction. I'm really excited to be with you today. So I was given the instructions to really share with you my career path and try to weave the science as describe my path. And I thought that this would be a good idea because, after all, once you watch my career path and you follow it, you will realize that if I could do this, each and every one of you could do what I've done, and you probably can do it even better. So we'll see. We'll do the journey and then we'll have questions. And I hope I can convince you if I could do it, you could do it. So I just want to maybe share at the beginning some of the key factors that have shaped my career. I would say if I was to put the most important ones, I would say mentorship, scholarship, the environment and support I got in that environment, wonderful collaborations, my trainees, and being a mentor. So these are the factors that I consider really have been very critical for my career. You've heard I come from Lebanon. I thought I'll show you a picture. I actually grew up in Beirut, right in this area here. This is the American University of Beirut. It's a beautiful city on the Mediterranean, and you see the snow-capped mountain around. And life was perfect when I was, 20 years old, started first year medical school, thought life was perfect. I did never expect it'll be any different. But the Civil War changed all that. And I ended up having to leave. My parents were worried for our safety, asked that I come just for the summer and return in October when school starts, but we couldn't return. It was too dangerous. And that's when I had to struggle to find a medical school that will take me 2 months late. And I'm really thankful for the dean at Meharry Medical College. Of all the medical schools I applied to, he was the only one who granted me an interview and said, you can start tomorrow. And that really, I consider a critical factor that shaped my careers. Now, on to mentorship. When I finished at Meharry, I applied to residencies in pediatrics, and I had super grades and high scores on everything. But I struggled. Many places did not want to look at me because probably I went to AUB and then came to the US, to medical school and so on. So it was really hard. I felt rejected from almost, again, every place I applied. But one person, the late Ralph Feigen, who's one of the country's most famous pediatricians, I would say he was the most famous 20th century, early 21st century pediatrician. He asked me what could he do to recruit me to Baylor. And I couldn't believe my ears that someone really believed in me. And I would say he changed the course of my life because I was facing many rejections at that time. So started pediatrics, loved cardiology, was going to be a pediatric cardiologist until I met Marvin Fishman, the chief of pediatric neurology, and I became fascinated by the brain. And he would tease me that the heart is not interesting. I would tell him the heart is far interesting than the brain. The whole month we were fighting. But when I finished the rotation, I really missed the brain and decided to become a pediatric neurologist, which I really enjoyed. But at some point, I found it very challenging. And this is Ashley, one of the children who I encountered that I found seeing them was really heart-wrenching. She was a healthy girl, did everything a 2-year-old will do, saying words, using her hands, riding a rocking horse, and then all of that stopped and her language disappeared. She stopped singing. She stopped running to her dad. Instead of using her hands, she would wring her hands. She developed features of autism, balance problems, and breathing problems. And to me, that was really devastating to imagine being born healthy and then something happens that robs you of all of that. This is where I say scholarship, digging deep in the literature really matters. Now, Ashley, I happened to see her the same month that this paper was published by Bengt Hagberg in Europe. It's an English paper in the American Annals of Neurology, but it was about European cases. No one has seen Rett cases here. And her pediatrician actually sent the paper with Ashley to tell us, do you think she has this disorder? And reading deeply, she had all the classic symptoms of that disorder. And all 35 cases were girls like her. So I went home and I kept thinking about her. And just by chance, a week later, seeing a child with cerebral palsy who walks into the room wringing her hands. What are the chances to see 2 girls with Rett syndrome in 1 week when no one has seen any in the United States? So that was serendipity. And after seeing 2, I asked the clinic volunteers, they're called the Bluebird Circle volunteers here, asked them to find me every chart of a girl with developmental disability and balance disorder and seizures. And I put some features of Rett. And they came back with 30 plus charts, and I was able to find 6 girls, was able to study them. And with the assistance of my attending faculty, I was still a first-year intern in neurology. So I have not really, I was still in training, but I was able to report on these girls, and doing spinal taps, we found they had reductions in biogenic amines in the spinal fluid. And this was published in the New England Journal of Medicine while I'm still in residency, but that was really life-changing in the sense because It's the first time pediatricians and neurologists in the USA are reading about this syndrome, about cases in the USA, and they started sending us their patients, and now you see girl after girl the same. And I was just really heartbroken about how the course is. You're born normal, then you lose milestone, but no degeneration. You stay like this for life. And it's that really that led me to decide to do genetic studies. So here I search for a mentor who will take me. Mind you, I've never done anything in a lab, so I was totally clueless about lab work. All I knew is I want to clone the Rett syndrome gene. And this was now July 1980, a little bit before July 85. And Art was willing to take me in his lab. I told him I want to work on Rett syndrome. He said, show me the families. I showed him these families. And as you can tell, it's only one girl in a family. And in 1985, there was no way to map a gene when it's just one in a family. So he said, no way. Of course, I was crushed. That's the whole reason I left clinical medicine to do science. But he said, you'll never find it. Remember then the genome wasn't mapped. In fact, when I started in his lab, PCR was not invented. So I had to really find another problem. And we went back and forth. And I finally settled on a very rare disease that affects balance and coordination called spinocerebellar ataxia type 1. Now, Art was happy with this because it's dominantly inherited. So you could map a gene if you had multiple affected individuals. And these individuals, they're born healthy, they're healthy till 40 years or so of age. Then they gradually, sadly, they lose their balance, become wheelchair bound and bedridden, and they die from choking. So it's a devastating disease. And I happened to find a family here north of Houston, and you'll see many, many affected individuals who were still alive at the time, so we could gather DNA to map that gene. But doing careful history, and this is where perhaps my background as a neurologist was helpful, revealed to me that there's something different about this family. Take that and couple it with a great environment where everybody was sharing information, everybody was sharing data at Baylor College of Medicine at the time. It was an exciting era. Fragile X syndrome just discovered, myotonic dystrophy syndrome just discovered. I became really aware that there's these expanded trinucleotide repeat that are causing these diseases. And Tom Caskey at the time, our chair, mentioned that in a seminar before the paper was published. So take it all together. I've got a great mentor who took me, Ralph Feigen. Marv turned me onto neurology. Art turned me onto, to study something other than Rett. And then, Tom Caskey shared the trinucleotide repeat story. All of that came together. And here I just want to highlight, all of them were really great mentors to me. And I think men can be great mentors. They just have to make they have to be cognizant of what's needed. And I feel each one of those men was very helpful in my career. So I transitioned after mapping the gene in Art's lab. He actually pushed me out of his lab after 3 years. I had a K grant, and he said, "You're ready to write an R1. Write an R1." I was very nervous. I didn't want to leave the nest, but wrote the R1 and I moved. And I insisted on being in the basic science building. They wanted to put me in the hospital building. I refused. I said, I'll take a bench in someone else's lab. I don't need an office. Just keep me in a very vibrant environment where the basic sciences are happening. And that's what I did. And I think having a bench and no office helped me because I was still doing experiments at the bench. And this is a point I want to really emphasize. When you start a lab, you are the best hands in that lab. And you're going to get some students. You'll never get the best postdocs in the country. You're going to get some, motivated, good postdocs, but perhaps not the most experienced. So therefore, you're going to be the best person. And by being in the lab all the time, you're training them to be the best they can be. You're also building the culture of the lab. And for me, it was really important to create a lab that had a culture of generosity. People share openly, help each other. And that's something happens when you're constantly in the lab and showing that in your own behavior. And at the beginning, that's another thing I advise young faculty. I only took graduate students. And the reason I say that, because I really feel huge responsibility towards postdoc. And when you're an assistant professor trying to make a name for yourself, it's really hard to hire many postdocs because you have to give them something to launch their careers. And if you're just starting, you don't have a lot of projects that you can carve away. So you're better off working with graduate students who go on to postdocs to keep some discoveries in your own lab that you build on. Then you will have a lot to farm out. So I started for 3 or so years purely with a graduate student. So I mentioned to you Fragile X was discovered, and we noticed in our and the myotonic dystrophy, we noticed in my family this disease is getting worse and worse as it goes from one generation to the other. And this is a hallmark of trinucleotide repeats. These are 3 bases of DNA that tend to expand from generation to the other. And having heard Tom Caskey describe the myotonic dystrophy gene, I immediately called Harry Orr. He and I were together trying to clone the gene, and we identified about 1 million base pair region. If you were to go gene by gene, this would have taken up probably 5 to 10 years. But I called him and said, what about if this is a triplet repeat? Let's just make oligos for all possible repeats, and let's divide the region of 1 million base pair in half. You take half, I take half. We search the region. We make sure there's 70 kilobases in overlap. And this is what we did. This was one of the most productive collaborations. And on the same day, we discovered the gene. We discovered it in his lab and in my lab, send each other faxes at the same time, not knowing the other one is sending a fax. And it turns out that the repeat was in the 70 kilobases of overlap, that we were careful not to miss any region within the gene. And in healthy people, this repeat is usually under 36, but in the patient, it goes between 39 and 82. Protein had absolutely no known function, so we called it ataxin-1 because it was the very first ataxia gene, and we knew nothing about its function. So that was the best we could do with it. We called it ataxin-1. And you'll see here, you remember the family, these are members of the Texas family. You'll see people with small repeats, right here, 40 had a very late age of onset, and those with big repeats are the ones who had onset as children. So this is the picture of Harry and I back in 1993 when we went to Naples at an ataxia meeting and presented our discovery. And after that, we both worked together. He worked with transgenic mice. I worked with knockout and knock-in mice. We showed that if you lose the gene, you don't get ataxia, but if you put a repeat within the endogenous locus as a knock-in, you get all the features of SCA1. We learned that this protein gets phosphorylated, and when bound by 43, it retards its degradation. And what the glutamine expansion does, it stabilizes this, it enhances this interaction. So basically, really, the disease is about slight accumulation of this protein. The mutation makes it slightly resist degradation. And we eventually found out some of the toxicity is mediated by its own partner, native partner, a transcription repressor called Capicua. And what we see in the disease, because you have more ataxin-1, you see more repression. So I'm summarizing a lot of data to really get you to where we are today about some of our conclusions. We discovered that although there's certain brain regions that are affected with this disease, such as the cerebellum and the brainstem, there are region-specific regulators of ataxin-1 levels. We discovered that Capicua drives disease in the Purkinje cells. If we prevent the interaction between ataxin-1 and Capicua, no matter how big the glutamine, we don't see toxicity. So this is beautiful. It tells you it's not just about the glutamine, it's about the glutamine in the context of the rest of the protein. So since the mutant protein accumulate, lowering it either with an ASO or a microRNA is a good strategy. But in work that I don't have time to show you, we found that if you eliminate the wild type protein in the context of disease, it aggravates disease. And that partial loss of this complex, the ataxin-1 Capicua complex in the hippocampus, increases BACE1 and Alzheimer's disease risk. So now you have a protein whose gain of function in the cerebellum, a complex capicuan ataxin, whose gain of function in the cerebellum causes degeneration, and whose loss of function in the hippocampus causes degeneration, just to give you a taste of how complicated biology is. So therefore, if you want to treat, really, you want to target mostly the hindbrain, the cerebellum and the brainstem. So just a word about collaboration, because Harry and I have been collaborating since 1988. And I would tell you, it's the best thing. It accelerates discovery. You maximize your resources, you build team spirit and moral support, because there are many days we both faced our challenges in the lab, and I would call him up and he'll cheer me up, and vice versa. Of course, you know, our chemistry was just right. We had the same philosophy, generosity, sharing, helping each other, protecting, you know, our trainees, so that each trainee will have their first author paper. So we agreed on everything. That made it easy for us to collaborate all these years. And you can see here our picture at the American Society of Human Genetics with this is Harry with my lab members. He even came all the way to Lebanon to meet my family. And of course, both my husband and his wife and all of us are good friends, but he's been to Lebanon to meet the family, and we continue to be best friends. So this has been one of the highlights I would consider of my career. Now, having studied ataxin-1, I quickly learned, as I mentioned to you, the importance of the level of the protein. Having too much ataxin-1 is what drives ataxia. Then that really opened my eyes to rethink about other more common degenerative diseases, such as Alzheimer and Parkinson. And here I'm going to share with you some of our work on tau, a major driver of Alzheimer's disease. This is a very nice piece of work that Chloe Chung, a current postdoc in the lab, generated these data. What you see here, we were curious, you know, tau levels seem to matter in Alzheimer. We were curious, do they matter in healthy aged mice? And tau, there are certain forms of tau that bind the microtubules that are not toxic, but when it comes off microtubules, it becomes phosphorylated and toxic. And there are antibodies that recognize both forms. So here you can see in young mice, this toxic form, as recognized by these antibodies, is not detectable, but you see it in 12-month-old mice, as you can see here. Whereas this form of tau that binds a microtubule and is not toxic, you don't see that at all. Here's another form of toxic tau that seems to also increase with age. So seeing that there are isoforms of tau that we know drive toxicity, and all these three isoforms accumulate in Alzheimer's disease. And now you're seeing them accumulating in healthy aging mice. So these mice are healthy, but just as they get old, they're having accumulation of these. Led us to say that perhaps if we can find genes that regulate tau levels, this would give us insight into its biology and reveal therapeutic targets. And Ji-Yeon Kim was at times a postdoc. She's now assistant professor, led this charge and collaborated with Ismail Al-Ramahi and Maria De Haro in the BOTAS lab, where she did her screens in cells to find regulators of tau levels. She used a fluorescent reporter, and infected the cells with short hairpins or guide RNAs, and find things that when you inhibit them, you'll either decrease or increase tau. And the BOTAS lab took flies that express tau and have degeneration, and then screened them using the equivalent genes to the human genes, and knock those genes one by one and see which ones make the disease better or worse. And after we did all that, we ended up with 260 genes of overlap. We validated many of these on the endogenous level, and even in vivo. All of these 16 genes were validated, and we tested 16, and all of them pretty much validated in vivo, 16 out of 19. So from there, we recently published a paper on the ubiquitin ligase of tau, and we found the degraders of that ligase, and we're pursuing that now to develop therapeutics. But most recently, we even identified a new kinase for tau. And just to show you an example of these data after we do these screens, here's a kinase, tyrosine kinase, that if you knock it down, you decrease tau in the mouse brain. So this is control and this is reduction. I see two different short hairpins, very nice reduction. This was surprising because there are hundreds and hundreds of labs that study tau phosphorylation. We could not imagine discovering a new kinase for tau, but we did. And to cut a long story short, it turns out there are 5 tyrosines on tau. 4 of them had been identified to have a dedicated kinase, but one of them, tyrosine 29, did not. And here you'll see that every time G expressed TYK2, she'll see phosphorylation. When any of those is mutated, the only time she loses it if the tyrosine 29 is not mutated. And then she was able to show that, again, if you express TYK2, you increase the protein levels, but if you mutate that tyrosine, the kinase doesn't work anymore and the protein is nicely degraded. And then she went into mice that have tauopathy and found that if you decrease TYK2, you decrease the pathology as shown here, decrease glia and astrocyte, and that's quantified here. And this is really exciting. I cannot tell you how many this is unpublished yet, but we presented it at the Keystone meeting. So many companies are reaching out to us because they have inhibitors, and we're really excited about this as a potential therapeutic in that disease. So we're trying to find our own small molecules to affect tau degradation, and this is another one that's independent. So while I'm doing all the work on SCA1, when we discovered ataxin-1, I got very disappointed that after basically 8 years of work to clone that gene, I got a gene of unknown function, ataxin-1. So I got really disappointed. So I went to my colleague, Hugo Bellin, who's a Drosophila geneticist at Baylor, and I said, I want a cool gene with some known function, because I want to work on something really that can do some molecular work with, unlike ataxin-1 I know nothing about. So he told me about this fly gene called atonal that was discovered by the Jan's lab, where it's in the hearing organ and the chordotonal organ for position sense. So I cloned the mouse homolog of that gene and subsequently made a knockout. And to my surprise, we found this gene is important for so many functions in the mammal. It's important for cerebellar granule cells, spinal interneurons, the Merkel cells that are for light touch, the inner ear hair cells in the cochlea and the vestibular system, the secretory lineage in the gut. And then from a very little area of expression in the hindbrain, it gives rise to multiple nuclei in the brainstem, shown here, either for proprioception sense, hearing, arousal, or breathing. I would have never imagined this one little gene will do that. So multiple papers, multiple fellows really worked on this, and it turned out to be very, very exciting. So this is an example where the cross-species work, although it was totally done for fun, ended up teaching us so much about the factor that's critical for the development of all these organs. So while I was spending all the time cloning SCA1, ataxin-1, and studying atonal, all of these papers coming out, thankfully, from the work on the ataxia and atonal, we were still struggling to find the Rett syndrome gene to no avail. Because remember, it's sporadic and no sequencing, so how are you going to find it? Eventually, we did have the hypothesis from the beginning that it has to be on the X because it's only girls, and Ruthie Amir has really, I would say, the postdoc who spent 3 years in my lab going gene by gene in this region of the X chromosome, where she was sequencing and characterizing gene by gene boundaries of the exons and sequencing the genes and checking them in patients. Eventually, she identified it in 1999 as the gene encoding methylcytosine binding protein 2. And what we learned from that is that in Rett, in the females, because they're mosaic, half of their cells have a normal protein and half lack the protein, and that gives you the full-blown picture of Rett syndrome. Whereas in males, when they lack the gene, they lack it in every cell, and it's pretty severe, and sadly, they die by 2 years of age. We also recently learned that milder mutations that still make a protein, but doesn't function at 100%, it may function at 80% or 70%, those will present with learning disability, autism, or any of these psychiatric symptoms. So there is a broad spectrum of the level of this, of the function based on the mutation. And one of the early experiments, and Collins, who was then graduate student, wanted to do is to add the gene to see if we can use gene therapy to treat this disease. And if you're going to do gene therapy, the first thing you're going to do is a positive control. And our positive control was to put a large piece of DNA that contains that gene. So that means the mouse had its own gene and an extra copy. But to our surprise, when we doubled the level of the protein, we found so many neurological abnormalities in this mouse. And it told us there must be humans with duplications of the gene. And this is indeed what we found. This is not by us. Linda Van Esch in Belgium, a year later, discovered this syndrome. But this is my first patient with that disorder, where they have duplications that span the MACP2 gene, and they have all the same features we see in the mice. So I'll summarize to you a lot of studies we've done in mice and humans, characterizing enhancers, mutations that change the level of the protein. At least these are in mice, that showed us that this is really a Goldilocks protein. You have to have it just right at 100%. If you totally lack it, it's fatal by 2 years of age. If you have it missing in half of the cells, as in Rett females, you have Rett. If you have it missing, is reduced to 50% in every cell, you also have a phenotype like Rett. But if you only decrease it by 30%, now you have features of autism and social behavior problems and anxiety. On the other side, if you increase it by 50%, you get, again, features of autism and behavior abnormality. And 200%, I just shared with you the duplication. We even have patients with triplications, and mice with triplications are very severe. Those will die in infancy, and the mice will die very early on. So it's amazing how much changing the level of this protein really matters. So with that, this really led us to think, it's hard sometimes at the time when we're thinking about this, it's hard to treat Rett because half the cells have normal level of the protein. So we're trying to think, if you're going to do gene therapy, you fix the deficient cells, but then you double it in the healthy cell. So we focused on the duplication, thinking, could we normalize the level of the protein in adult mice and rescue their features? And this is, and we decided to do it genetically, which we showed you can. And this is work done by Hezi Steinberg, who at the time was a postdoc. Then we collaborated with Ionis Pharmaceutical and asked if we use antisense oligos, would we normalize the protein and could that reverse the disease? And this Hezi and Yanyao, she was a graduate student at the time, collaborated to do this. And for most of you probably know what antisense oligos do. They bind to the RNA, and then the DNA RNA duplex, heteroduplex, will be digested by RNase H, degrading the RNA. So you decrease the protein production. And this is what we found in the duplication mice. We found, for example, here's a healthy mouse active. Here's a duplication mouse, not active, but a duplication mouse that received the ASO and normalized MACP2 level comes back to be active again. Here's, again, healthy transgenic ASO treated in these various parameters of behaviors. We even took mice that are very old, 8 to 9 months of age, having seizures every day. And we found if we treat them, you can see here they're having all this electrographic seizures, they're clinically having seizures. We normalize the seizures disappear and they do much better. So in summary, we learned, we repeated the studies, we created humanized mice and showed you can titrate the ASO to where you decrease it to the desired level because you don't want to overshoot. And we learned that the RNA normalizes first, then the protein normalizes, then the targets of MACP2. Many genes change when MACP2 levels go up or down, and then we see the behavior rescue. That takes a little bit of a lag time. So I'm very happy to share with you that yesterday, Ionis Pharmaceutical put on a press release announcing the molecule Ionis 440, which is derived from the studies. We gave you this preclinical proof of concept, and they will do the clinical trial for these patients in 2024. So we're very excited about that because it took really 10 years of preclinical proof of concept work to get us to that point. And we're very excited for the patients because these boys unfortunately die by the third decade. They suffer so much seizures and neurologic problems. So hopefully we can help them now. And I just, in the last 5 minutes, I want to share with you a new unpublished story that a postdoc in the lab, Samir Bhajakar, has done to address two questions that have always intrigued us. Whenever we looked in the RNA from mice that either lack or overexpress MACP2, we found so many changes. Many genes go down in the knockout and some go up and vice versa. So you don't know what is primary and what's secondary. And you worry that maybe it's because the neurons are sick, you're seeing a lot of changes due to sick neurons. So we wanted to really define the earliest molecular changes upon the loss of the protein. We also, as I mentioned to you in humans, it took time for the disease to manifest in childhood. We also did an adult knockout. It also takes time for the disease to manifest. So why does it take time? And if you recall my ASO treatment slide in the duplication, they are very sick mice. When we treat them, we normalize the protein by 2 weeks. But again, it takes almost 2 months for the behavior to normalize. So both times, both ways, it takes time. Why is that? So to address this question, Samir did an adult knockout of this gene, but he practically did 6 adult knockouts. He would do an adult knockout and will harvest RNA, protein, and assess behavior and weight at 1 week after the knockout, then 2 weeks, then 3, then 4, then 6, and 8. Each of those is a different knockout, obviously, but you're waiting each time to see what are the earliest changes, the next, and so on and so forth. So for that, he evaluated gene expression, the histone marks, physiology, and behavior. And what we learned is that the RNA immediately goes down. When you knock this gene in the adult, it immediately goes down. But the protein lags down a little bit. So only by, say, 9 days or so, it's about 50% gone. By 2 weeks, maybe 60, 70% gone. And it takes really 3 to 4 weeks for it to be completely gone. So what happens now to gene expression? Well, it starts really early on. Almost from the time you lose 30% of the protein, you start seeing gene expression changes, and it continues and continues. And some of the things that were changed early on will just now be changed to a greater degree. And then he asked, how about histone marks? And to our surprise, even at 1 week, this acetylated K27 histone 3 mark, which is a mark of active gene expression, you can see in those genes that are downregulated, even at 1 week, it's decreased. And for those that are upregulated, it changes a little bit later. So this was quite interesting because, as you can see here, the chromatin change was, again, almost as early as the first reduction of MACP2. Then we looked at physiology. And here you see only at 8 weeks, we detected physiology. At 4 weeks, it was normal. We now are recording at 6 weeks, it's minimal change. So this again tells us chromatin and gene expression happened first. And body weight, it's from the hypothalamus. All of this other work is in the hippocampus. Physiology follows, and then behavior follows. So this has been really helping now to understand the disease course. So, let's see here. So now I want to sort of really conclude to tell you about some of the things I've learned along the way. As you can tell in my slide, I always bring mentorship just because it's my favorite part of the job. So what's the best part? the best part is you're watching intellectual growth in real time. You're watching these brilliant minds learning and growing and becoming independent in their thinking. And they're the ones starting to bring great ideas. But you're always being refreshed. I always feel refreshed because I'm a mentor. By hanging out with my students and fellows, I am learning from them as much as I'm teaching them. I'm learning new things. Certainly, many of the things of the modern world, they teach me that's for sure. And we really build amazing relationships. The other really important part for me, I realize I'm only one person and I can only do so much. So when I one day no longer have a lab and no longer do science, I want to be sure the science is in good hands. And the more we successfully mentor people, the more likely for that to happen. And it'll multiply the effect that I dream to have. If I was to do one thing by myself, that's not going to be enough. But right now we have 107 graduates. So since I guess you got my CV, few more graduated and moved on to faculty positions and other positions. So we have 107 graduates of the lab. So I'm very proud of that multiplier effect. And I thought it'll be nice to show you pictures, different stages, different times. This is Ruthie. She deserves a very big picture because she worked tirelessly to find that gene. And these are the various lab members. So there are a few things that are important to talk about a little bit, I think, that I feel helped me in my career. First and foremost, I would say naivety, because I cannot tell you how many people thought it was totally stupid to go after the Rett syndrome gene, for example, sporadic disease, wasting your time, etc. But I don't know. I mean, I think my naivety and willingness to take risk probably played a big factor. I walked into the lab optimistic that I'm going to find the gene. Now, in hindsight, if I knew it'll take 16 years, I don't know that I would have committed to that. But luckily, I was naive, truly. So I didn't know. So you keep at it till it happens. I think definitely the passion helps. I would say it was the passion that drove me from the clinic to the lab. It was the passion that pushed me to do these 10 years of research to help us find something for the duplication. And we're continuing to work now on Rett syndrome. I think perseverance and resilience are important. Patience, one has to be patient because science isn't always cranking out discoveries. So sometimes it gives you a small piece of knowledge that's helpful, but it's not that exciting. So you just have to be patient and things will fall into place. And definitely, with all my parents in Lebanon and nobody here, William and I raising a family, my, family unit was very important. I remember vividly every time I would leave the lab after working so hard for a week and the whole experiment gave me negative results. I mean, I'm probably my postdoc and the first few years of my lab were all negative data. So I'm the queen of negative data in those era. So, what helped me is I'm going to go home and I'm going to see the faces of my children. And that's really helped me. So connecting with friends, partners, families, that really makes a difference. So I feel that helped keep the balance in my life. And I thought you might enjoy seeing our son and daughter and his wife, Zena and Tyler. And now we have 3 little munchkins, Rula's kids and Sienna's Anthony. So these really give me a lot of joy. And with that, I want to thank first and foremost all the patients and their families and my incredible environment at Baylor College of Medicine. I think the collaborative environment here is unparalleled, really unparalleled. I visited many places. I feel so fortunate to have grown in this place. And of course, the Howard Hughes Institute funding, and then all of our funders, JPB for Alzheimer, Rett syndrome research and duplication, and the NIH and IONIS. And this is where my lab is, right here. So with that, I'm going to stop sharing. And let's see, how do I stop sharing? Yes. Happy to take questions. And we have good time for questions.
Yes, We do. Thank you so much, Dr. Zogby, for your inspiring lecture on science and career. Your journey in both research and mentorship is truly remarkable. And it's so inspiring to see years of hard work actually go into the next step to like betterment now that it's going to trial. So that's like clinical trial. That's really inspiring to see this entire journey go to fulfillment. So I'd like to remind the audience to please put your questions for Dr. Zogby in the Q&A section. We are looking forward to discussing both scientific and career-based topics, and we'll try to make it through as many questions as possible. But I will first begin with some of the questions that were submitted by our selected fellows and associates, and then we'll take the audience questions. So one of the first questions that everyone had was balancing the demands of a successful career with personal life can be challenging. What were your turning points? What were some of the turning points in your life when you deliberately pivoted towards either investing more time and energy to your career or life? And looking back, was that the right choice? Or would you have made it? What other suggestions do you have?
Well, thank you for giving me time to answer this question. This is such an important question, and I didn't really address it in my talk, so this is a perfect time to address it. I'll be very honest with you and tell you, it's not that I put sometimes more emphasis on life versus work to balance things. I consider it all life. It's all life. And my work and family are intertwined. So I'll share with you what I did that I think helped. And let me say one thing. When I was doing it, I didn't know it's going to work. I would confess that for 10 years of my life, from the time I was a postdoc through assistant professor and associate professor, because my kids were born when I was a postdoc, all those 10 years, I was petrified. I thought my kids are going to turn on to be something bad because I wasn't the mother who's manning the, mother's desk in the school. I wasn't the mother who's helping in the school decorate the school. I wasn't the one who's always doing things and always home there. So that was very petrifying, and I didn't have a role model. I mean, now we're so fortunate. There's so many you can look up to and ask these questions. So I was just doing it and just worried. And what I did, though, I drew boundaries. And the boundaries are, if the kids need me, they're number one. I brought them to this world. If they're sick, if they need attention, I have to drop everything. I have to repeat the experiment. I go home and take care of them if they're sick. That was number one rule. They come first. Number two rule, I was very organized and a big planner. I would plan things on the weekend, prepare for the whole week, whether it's the food that we're going to eat the rest of the week, or whether it is the, schedule that I'm going to do in the lab. Sunday was my big cooking day, planning day. I did all that. I did call on help. You cannot do everything. No one is so super that they can be a mother, a wife, a great scientist, and a physician. At some point, I went back to the clinic. I was doing 20% clinical work. So there I hired a babysitter who would come during the day between 7:30 and 5:30 to be with the kids. So I put structure in my life. She would arrive at 7:30. I will go to the lab. 5:15, I will leave the lab and I would be with her. I'm with the kids, undivided attention to them till they go to sleep. By 8 o'clock, they're asleep. My husband and I will eat dinner. I'll go back to the lab and plate my Cosmid library, my phage library, start my Southern blot. I did things like that helped me always get a head start. Sometimes it wasn't every evening that I went back, but many evenings I went back where I put in an hour and it helps me get a head start on the next day. On the weekend, my husband and I will split the day because he was a fellow too in cardiology. So I would take the morning, he'll take the afternoon. Sometimes my daughter will come with me to the lab. Things were a little bit more loose back then. You could bring them to the lab. Now you can't. But that's what I did. I really sort of weaved it all in, but I did call on help. I didn't have time to clean the house. I paid someone to clean the house. Literally, my whole salary as an assistant professor, as a postdoc, senior professor, went to all the help between, the babysitter, the person who cleaned the house, the person who cut the grass, and the groceries. That's it. That's the whole salary went to that. But that's an investment in your career. And I think that, and when you treat people who are caring for your kids well, they will also be part of your family to this day. They're part of my family to this day. So I think I was also always looking for efficient things. I would never go grocery shopping on Saturday or Sunday because it's going to take you twice as long. I would go Tuesday. The produce are still fresh Tuesday night. Nobody's in the store. I whiz through the store in 30 minutes and I'm out. So find things that increase your efficiency. But really, it's all about planning and being organized. And if you want to make a spaghetti sauce, whether you make this much or that much, it's the same. You're dirtying a pot, having to have all these ingredients. So just do more, freeze, put away. This was my modus operandi to increase my efficiency. Came the time when they were about 10, 11 years old, because I was not in their face all the time. They always loved me. While all adolescents rebel on their parents, our kids really loved being with us. So all the other parents now that I was so envious on, and who sometimes look down at me because I am missing in action in the lab, come to me and say, well, can you advise us? We're finding this problem with our kids and so on. Because I gave them space, they had structure in their life and they really loved us. They never got tired of us. And I think both of them work, their spouses work, which tells you that if they were traumatized by the experience, that wouldn't happen. So I share all this with you because I didn't know this at the time. I felt so guilty. I was so scared. But now that I know better, I share with you so that you don't feel bad and you don't feel guilty. They will be fine. But as I said, they are a priority. I may not have made it to all soccer practices, but I sure made it to every game. Right? So you go for what matters. And I think that's what's really important.
Thank you so much for sharing all of that. It's both inspiring and also really relatable, I think, to many people in our audience, especially to me and Sohini as well. We can definitely feel that. So I just want to follow up with one more question from our fellows that also sort of combines a couple that we see in the chat right now. So we understand that you started a fellowship or an endowment fund specifically to help postdocs transition to faculty careers. We'd love to hear you speak a little bit more about what motivated you to set this up and really how important do you think it is for faculty, especially those at your level, to really use their influence to change the system, but also to then sort of help the postdocs identify sort of when they're ready to move on to the next step?
Thank you, Michael. This is really a good question. My inspiration to do that was really simple. When I started getting these big prizes, I was like struggling. What do I do with them? It's not only me, right? It's like the people who came ahead of me, who mentored me. It's my collaborators. It's my alma maters in different institutions. And it's my trainees. So you honor all these others with special things, and there are, things in their honor and their endowment at all these institutions because they're important to me, obviously. But then remains the trainees. I couldn't figure out what can I do for the trainees to honor, because I consider any prize to me is a prize to all of us, right? And it's not one person of my trainees, it's everybody who's come along, because that's how science is, you build. So then I thought, well, the only way to really honor the trainees is to do something that's meaningful for trainees. And what is meaningful? Many trainees have great ideas when they're like third, fourth year fellows and about to go, they may take something from the mentor's lab, but they may have a very brilliant idea that they want to start. Or even if they're taking something, if they had a mouse, if they made the mouse, or they did a, RNA-seq experiment, what have you, some experiment, it'll just help them when they start their job, be more competitive when they go on the job market, and second, have more data for a grant. So I thought, well, what would help is perhaps if there's a pot of money, if you create an endowment and from that some money comes up and they apply for that, and there's a pot of money that helps the fellows at that stage to take that money. So this way the mentor is not feeling the pinch I mean, I'm fortunate. My lab, we are a huge lab. I can afford let my postdoc make any mouse they want and take it with them. But smaller labs, they can't always have that. If you're solely dependent on the NIH, sometimes that's not easy. So this gives an attention to those postdoc where they can pursue a bright idea, and hopefully that will help them launch their career. So that's really, the inspiration is gratitude for my trainees over the years and recognition, and to do something in their honor. And it's something practical.
That is such a lovely gesture in terms of like showing how wonderful a mentor you are. In that similar line, we have a question from the audience. I'm sorry if I'm pronouncing the name wrong, Dr. Utecho. He says, what specific characteristics or approaches have made your male mentors effective in supporting and mentoring women in science?
It's different for different people. I can tell you that. For Ralph, I think Ralph is one of those incredible human beings who looks at a person in front of him, doesn't let anything bias him in any way, shape, or form. And therefore, he treats you for what you are. And I think that's very special. That's what makes him. So it doesn't matter your gender, it doesn't matter your color, it doesn't matter which university you went to, whether it's an Ivy League or not, it didn't matter to him. He looked at you as a person and empowered you. So that's what made him really special. I think Mark Fishman really, what made him special is he just keep beating me to love the brain. So I would say that was his distinguishing feature. Art Baudet was different in that he is a tough mentor on everyone. He demanded a lot of us, and he pushed us to be the best we can be. But the beautiful thing about him is it doesn't matter if you're a woman or a man. He pushed you to your best potential, treated everybody equally. And when you feel you're being treated equally and pushed to your potential, then it feels okay. So that's what I'm saying. They were all different in their styles. None of them, I would say, necessarily, or particularly, I mean, for the lab art, he didn't necessarily pay as much attention to my family life needs, and I didn't engage him in that. But he never, ever made me feel that I need to do X, Y, and Z. He just basically set the expectations, and he set them for everybody, and he just pushed you to be the best you can be. And that's part of it inspires confidence. I mean, you underestimate it because I was totally not confident in myself, So it takes a while to gain that.
Thank you again for sharing sort of what your mentors have done for you. And it's very clear that you do the same thing also for your mentees as well. We have another question from the audience from Dr. Mudumbi. Could you provide some more details of how you worked out protecting your trainees? For example, making sure they got first author papers when collaborating with more senior faculty.
Very good question. It was very simple. That's where, for a collaboration to work, there has to be absolute transparency, honesty, respect of boundaries. Those are critical criteria. So Harry and I, from day one, we told each other everything. Whatever I'm working with, I tell him. Whatever he's working on, he tells me. When I write an R01, I tell him the aims of my R01. When he writes one, he tells me his aims, and we share, and so on and so forth. Then what happens is a postdoc in my lab is working on a particular experiment. I tell him that, and he knows that. His lab might do something that helps our experiment, but his postdoc will work on a different experiment in a slightly different domain, and my fellow might help that person. So as long as we give everybody a project that there's theirs, but they could exchange data and information. They could have an author from that lab on them and vice versa. So all you have to do is go through 3 or 4 years right now of authorship from our labs. you'll see it. Like we recently had 2 papers back to back. My graduate student, first author on my paper, his is on it, his graduating first on his paper, and the 2 papers back to back. Sometimes we have 2 co-first authors, but we've always worked it out because we respected the boundaries and we never hid anything. That's really critical. If I didn't tell him everything I'm doing, right? If I only told him half of what I'm doing, and Wendy, one of his students, come up with an idea, and it's in that half I never really revealed, that creates conflict. But since I tell him everything and he tells me everything, it's all out there. We know how to navigate the pieces. That's how we did it.
Thank you, Dr. Zogby, for taking time to share your expertise with us. We are at the hour now, so with this I believe I speak for all of us here when I say that we are not just walking away with knowledge, but also with inspiration. Dr. Zogby has set an example of excellence in research and a passion for making a difference in the world, and I'm sure her influence will continue to motivate us in our respective fields. And with this, we would like to really thank you for all your time and sharing everything with us. Thanks a lot.
Thank you so much. Such a pleasure. You know, you all made me feel so refreshed today. Although I woke up feeling a little bit sick this morning, I feel so much better after connecting with you. Thank you so much. Really appreciate it.
It's our honor to have you join us. So thank you so much again. And now we'd like to invite our outreach committee co-chairs for their closing remarks.
Good evening, everyone. We have had quite an enriching second day of ISFS. We heard from our faculty panel on the realities of being a PI. We learned about the exciting research of our associates, and we were inspired by the research endeavors of our fellows in topics ranging from biochemical and biophysical processes to communications of cells and beyond. We were blessed to have Dr. Zogby discuss the molecular mechanisms of neurodevelopment and neurodegeneration. The day is not over yet. Please stick with us this evening for networking sessions. Now, before we get to the networking sessions, we wanted to take this time to introduce ourselves and our committee. My name is Jennifer Sun. I'm a presidential postdoctoral research fellow at Rutgers University, where I'm initiating my research lab as part of my unique tenure track path to professorship position.
And I am Tigis Tamer. I'm a postdoctoral associate and NIGMS Mosaic Scholar at the Cook Institute at MIT. We are co-chairs of the Outreach Committee. Our committee of postdocs and volunteers have been working tirelessly to make sure that we highlight our fellows, make sure we provide information on opportunities and events, and that we champion your achievements and connect you to institutions seeking faculty applications. Going forward, please continue to monitor the website and the Twitter account so that we can keep you abreast of all of the job opportunities and also celebrate your success as you move forward in your career. And we wish to also take this time to thank our postdoc volunteers in all of the different committees that have put the energy and time to prepare a lot of this program information, professional development opportunities, and especially our committee who have done an amazing job making sure that there's proper lines of communication and everybody feels seen and observed. And that the mission of ISFS succeeds. And this was a heroic effort, which involved constant crosstalk between all of these committees. So we also want to acknowledge the co-chairs of the program planning and professional development committees. And with that, we thank all of you for being here for ISFS Day 2. And we wish you an enriching evening of networking. And we encourage you to stay and meet the fellows or stay in the public networking session. Right, thank you so much.